In this second part of Chapter 4, we're going to be looking at different ways in which molecules can move across the plasma membrane and different ways that cells can communicate with one another. So there are three main mechanisms through which molecules can pass through the cell membrane: passive transport, facilitated transport, and active transport. Passive transport is when there's no energy involved and simple osmosis or diffusion is going to occur. Facilitated transport will also not require any energy, but is going to involve a transport protein helping a larger molecule pass through the membrane. And with active transport, energy is going to be required in order to move the molecule through the cell membrane. So as we look at passive transport, there are two basic types, and that is diffusion and osmosis. Items moving down their concentration gradient, moving from an area of high concentration to low concentration, is going to dominate in these two. So if we look at diffusion, what we start out with is some form of solvent, an item that is going to be involved in dissolving an item. So remember we said that water is a universal solvent. Most things will dissolve into it. Into this we put a solute. The example that you see here is colored water being placed into just plain water. Initially, there is a high concentration of the colored water in one central area. Over time, this is going to diffuse. The molecules are going to spread out until they're evenly distributed and an equilibrium is reached. And in the image below, you can see the high concentration of the solute within the solvent. You can see it slowly having the molecules spread out, and then eventually an equilibrium is reached, and there's a fairly uniform distribution of the solute within the solvent. So with simple diffusion and osmosis is that same sort of diffusion, same sort of process, but when water is the item that is doing the moving. With simple diffusion, molecules are going to pass directly through the plasma membrane. They're not going to need any assistance, and they're not going to need any energy. With facilitated diffusion, the molecules are still going to be moving across the plasma membrane. They're still not going to require any input of energy, but they tend to be too large or too oddly shaped to fit through the plasma membrane by themselves, and are going to rely on one of the transport proteins or carrier proteins to enable passage of the molecule through the plasma membrane. And of course, defects in these transport proteins can cause problems. They can cause health issues and even death. Cystinuria is a disorder in which the proteins are not able to move cysteine, which is one of the amino acids, across the barrier. And what happens is you get a buildup of these proteins on the inside of your kidneys, and that is how a kidney stone is formed. Now, again, when water is doing the movement, when passive diffusion is taking place and water is involved in the transport, instead of calling it diffusion, we call it osmosis. So you can see in the first picture we have an animal cell, specifically a red blood cell on top, and we have just a generalized plant cell on the bottom. When there is a balance, this is called an isotonic solution. You have an equal flow of water into and flow of water out of the cell, so they balance out. In a hypotonic solution, water is going to diffuse into the cell through the membrane to the inside where the intracellular fluid is. In an animal cell, unfortunately, they cannot sustain too much of this and they will tend to burst. For a plant cell, since they have a cell wall which is going to help maintain rigidity, the cell will expand, and that's what we call a turgid condition. And in a hypertonic solution, water is going to exit, it's going to leave out of the cell, and again it can cause rupturing in an animal cell, and in a plant cell it will cause the cells to shrivel, and that's when we get plants that wilt. So as we look at an example of this in a situation with celery that's placed into distilled water, into a 1% salt solution, and into a 10% salt solution, we can see that after a few moments, in the distilled water, water rushes into the celery. That is a hypotonic solution, and the celery itself swells and becomes turgid. In the 1% salt solution, that's about the same as the natural condition for celery. It stays pretty much the same. There's no change in texture, and water is not going in, so there's no change in weight. But in a 10% salt solution, the water goes rushing out of the celery. It becomes wilted and withered, and the weight decreases. And we rely on this for some medical situations. If you're suffering from constipation, we'll tend to drink milk with magnesium or magnesium salts. And what this does is increases the salinity within the intestines and causes a hypotonic situation where water will come moving into the intestines, and this will help relieve the constipation. And so with osmosis, it doesn't matter what the solute is, if it's a salt or some other particle that you're placing in the water to dissolve it. As long as the total concentration is different, water is going to move. And since this is osmosis, it's a form of passive diffusion. It does not take any energy for it to do so. So think about this for a moment, pause the video, and when you're ready, come back. And here's an answer. This is not the correct answer, but it is a correct answer. If you're uncertain, as always, feel free to Sally or email a question. Now, molecules can't always just spontaneously move. Diffusion and osmosis can't always happen, nor can facilitated transport. Sometimes the particles are too large, or you need them to move up their gradient. You need them to move into an area where they're already in a higher concentration, and that's going to involve energy, and that's what signifies active transport. So there are two basic types of active transport, primary and secondary. With primary active transport, the movement of the molecules is again going to require energy, and it's generally used when you need to move them against their concentration gradient rather than down. So if you are going to need to pump more acids, more H+ ions into the stomach when you are digesting foods, you're going to require some energy. You're going to get more energy out of your food than you put into pushing particles into the stomach against their concentration gradient, but it does take energy. And in secondary active transport, with secondary active transport, the body is going to simultaneously move one molecule against its concentration gradient while letting another flow down its gradient. And in this case, sodium is going to move down its concentration gradient, whereas potassium, which is needed in high concentrations inside of the cell, is going to be pushed against its gradient. So we take some energy to force the potassium to where there already is a greater concentration, but some of that is going to be assuaged by allowing the sodium to move just down its normal concentration gradient. So take a moment to answer these, and when you're done, advance the video to see the answers. So hopefully you got those, but if you have any questions, feel free to use my office hours or any of the other forms of communication available to you to ask for help. And some molecules just cannot pass through, whether it's even actively using energy, they just can't get through the plasma membrane. And in these cases, we're going to rely on endo bringing into or exocytosis, in which we're going to use vesicles. We're going to actually kind of pinch off parts of the plasma membrane to allow these molecules to move through. So with phagocytosis, this is a type of endocytosis in which the cells engulf a larger particle. And on the right here, you see a phagocyte consuming a bacterium. So the plasma membrane is going to form this pocket-like vesicle, and you can see one of the receptor proteins on the left-hand side is binding to the particle. This initiates the downward motion towards the cell of that portion of the plasma membrane and an upward motion of surrounding portions of the plasma membrane to move up and literally swallow up this particle until it's completely encased within the plasma membrane, which will then shut itself off and bring the particle to the inside of the cell. With pinocytosis, it's pretty much the exact same process, but instead of being a solid, the particle that's binding to the plasma membrane is a liquid. So if you're bringing in any sort of dissolved particle or liquid, it's pinocytosis instead of phagocytosis, bringing that substance into the cell. With receptor-mediated endocytosis, there's going to actually be a receptor that is binding to the molecule that's moving in, and this is the case with LDL, or the low-density lipoproteins. You've probably heard that as one of the two forms of cholesterol, LDL and HDL. This is the bad one, and usually what the body will do is it will bind with the LDL, bring this into the protein, and will enable the LDL itself to be broken down and used to make molecules like estrogen or testosterone. Of course, sometimes people don't have functioning LDL receptor proteins, and they're not able to break down the LDL in the manner that they should. And that can, of course, result in various forms of cardiovascular disease. So you can see the healthy artery on the left, which is partially blocked by plaque, or this buildup of that LDL, and completely blocked, which can lead to either a heart attack or stroke. So kind of think about a way to treat this, and that's something that we're going to discuss in class. And of course, if you can move particles into the cell, you can also move them out, and that would be the process of exocytosis, which is pretty much going to work the same way, but instead of having the receptor molecules on the outside surface of the plasma membrane, they're going to be on the inside, so that when there is something like a neurotransmitter on the inside portion, it can have a vesicle created around it to bring it out of the cell. And so think about this one, and again, when you are ready for the answer, move to the next slide. And hopefully you realize, since it was leaving, you do have exocytosis. It's exiting the cell. Okay, and then there are connections between the cells which are going to hold them in place. Essentially, they're fasteners, like having glue or having Velcro attaching the cells. They're going to connect them to one another and enable them to communicate. So we have a few basic types. We're going to focus on just the three: the gap junctions, which are essential for allowing communication but also act as a passageway. You're going to see these in the heart as well as with, well, we'll get to those in Bio 102. And the tight junctions, these are going to form a watertight barrier. They're going to act almost like biological caulking, and these are critical in areas like the stomach and the small intestines, where you can't risk having some of these acids leave the contained area that they're in, like the intestines or stomach. And desmosomes act like kind of like Velcro fasteners, and these are very common in tissues lining cavities, so in the esophagus and other areas in the body. So your skin is waterproof. Think about this question and move to the answer when you're ready. And a specialized form of connection is a plasmodesmata. This is only found in plant cells. These are tube-like channels that connect the cells. You can see them in sort of the dark gray on this lower image, where you have an individual cell with this plasmodesmata around it, which really connects this one cell to all of the surrounding cells, allowing a very easy transport and communication as well, and can almost allow you to think about a plant as one ginormous cell, so it's pretty interesting. And in the last section, the last video, we're going to be looking at the organelles within eukaryotic cells.